for Duchenne, where are we in the clinic, and what's coming next. Now, you will also learn a bit about anatomy, genetics, immunology, and virology. So there's also an educational aspect to this. For those who don't want to listen to me for 30 minutes, this is a summary of my talk. There's no approved gene therapy for Duchenne in the UK yet. But we have a very good understanding of the concept, are becoming more familiar with the safety challenges and their management, and aim to further improve and confirm the efficacy through clinical trials and real-world data. This is where the things stand. This is what I will talk about. So the concept of gene therapy. Now, when I give the diagnosis of a genetic disease, Duchenne muscular dystrophy, I always tell patients, you don't need to know a lot about genetics, but you should understand the genetic dogma that a gene, or the entirety of genes, our genome, is like a cookbook. And within this cookbook, there are recipes, and the DNA is transcribed into RNA, the recipe, and that recipe is used to make a protein or the dish that is a substance that's important for the function of a cell. And depending on where this recipe is in the tissue, in which tissue, for example, in muscle, and if the protein is dystrophin, this is an important substance to maintain integrity of the muscle fiber during cycles of contraction and relaxation. And if you have a genetic disease, this means that the cell cannot produce the recipe for that tissue, it cannot make the protein, and if that's missing in that tissue, this can result in, for example, muscular dystrophy if you don't have any dystrophin. And then patients say to me, well, can't you just replace the faulty gene with a healthy gene? You know, it's a very easy concept to understand. Because then you have the recipe again, then you can make the protein, and then you have healthy muscle. And so gene therapy is a very simple concept compared to antisense oligonucleotides. But how do you get the healthy gene into the cells? That's the challenge a bit. Now, this is where scientists have used viruses, because since COVID, everyone is a virologist now, and you probably know that viruses aren't living organisms. They have to use host cells, and they provide the host cell with genetic information to make more viruses. But they can't live and replicate without a host cell. And then the question was, well, can we use viruses to transport our gene of interest into the cell, and the cell makes the recipe and the protein? And the most popular virus that is used in all of the gene therapy trials actually for Duchenne muscular dystrophy currently is called adeno-associated virus, or AAV. It's a small and simple virus, which is great because it elicits only a weak immune response. Many of you here, probably 20 to 30%, will have been affected with several AAV viruses as well, but they have never shown to cause any disease in humans. Now, the genetic information of the virus is taken out, and we'll replace it with our gene of interest. And we also need to have a promoter. And a promoter is like an ignition to activate the gene in that tissue where you want it to be active. Okay? So I will use the term promoter. It's like activating the gene in the tissue of interest. And viruses, once they have their gene of interest in it, they dock to so-called receptors at the surface of a cell, and then they are incorporated into the cell, and they deliver the recipe, mRNA, in the nucleus, and then the protein, our dish, is produced. Now, they have a GPS system depending on the virus. There are many different strains of adeno-associated viruses. For gene therapy in Duchenne, we mainly use AAV8 and 9. And these different viruses bind to different receptors, and therefore they have an affinity to a specific tissue. In terms of the concept for gene therapy, you also need to understand there is a challenge with the immune system. Now, this article here that was published by Amy Rosenberg as the first author, she's from the FDA. She explained that if in a muscle fiber without dystrophin, you have damage, and that also triggers an immune response. And there's a so-called innate, immediate, first line of defense immune response. And then there is a so-called adaptive, which is a memory. This can happen in response to damage or in response to a virus. And all of you, of course, know if you have a vaccination, some people say after the vaccination, in the first few hours even or days, I can feel that there is an inflammation, and that's the innate immune response. And then the principle is that the adaptive immune response memorizes this, and if the virus attacks you again, you have a good defense system. Now the challenge is with viruses, if you overload the system, that the immune system will respond. But the challenge for Duchenne is also if you reproduce your protein, dystrophin, then the immune system may have never seen dystrophin before and considered is to be foreign and attacks it as well. These are challenges. So we have a great concept. We know safety first, but still what's frustrating for families is we have talked about gene therapy for decades. Interestingly, the first AAV vectors were described more than 40 years ago. And the first AAV clinical trials also, took place about 30 years ago, even using dystrophin as well. The first mice with dystrophin were injected more than 20 years ago. We still don't have any approved drug in the UK. Meanwhile, for another disease, spinal muscular atrophy, everyone has heard that there's an amazing gene therapy. So one of the questions is, what are the challenges in Duchenne muscular dystrophy? And I thought I'll focus on the challenges, comparing it to spinal muscular atrophy. Because everyone talks about spinal muscular atrophy, how amazing it is. Why does it not work in Duchenne muscular dystrophy? So spinal muscular atrophy type 1, these are babies that also have muscle weakness, severe, profound muscle weakness. They're so severely affected that they would normally, most of them, die within the first 2 years of life. And there are about 40 to 50 new babies born in the UK, about 70 males with Duchenne. So Duchenne is a bit more frequent, but these are a bit like the flagship diseases for experts that work in the neuromuscular field. And when we talk about neuromuscular conditions, this is the anatomy lecture part. You know that our motor function is controlled through the motor cortex, and then there's a first motor neuron. But neuromuscular diseases are caused by changes in the so-called motor unit, the second motor neuron, or the muscle itself. Duchenne, of course, affects the muscle itself, where spinal muscular atrophy affects the second motor neuron, so the nerve that actually activates the muscle. There's one really important difference, and many differences, but one that is very important for gene therapy is that neurons do not regenerate. We are born with a definite number of neurons, and they don't get more, and if they die, they're gone, they're not regenerating, which is bad for the central nervous system if we lose neurons. But it's good for gene therapy, because once you have a gene of interest in the motor neuron, it stays in there. And the motor neuron has only one so-called nucleus, and I said this is where the virus delivers the recipe, the gene of interest. Whereas in muscle is a tissue that can regenerate, so we can lose muscle fibers and generate new ones as well. And that's not ideal for gene therapy because it could dilute the efficacy of gene therapy. And we need to target many, many nuclei within the muscle, which is also a challenge. So muscle is more challenging than motor neurons. There's also an important aspect about the immune system. And I mentioned that in Duchenne, because of the damage, there's already a very active immune system. This is why we use steroids, by the way, to suppress it. The babies are injected with a lower absolute volume or dosage of vectors compared to someone who weighs maybe 15 kilograms or more. So it's a bigger challenge for the immune system to deal with more viral load. If you inject a newborn, and that's what we are aiming for in spinal muscular atrophy, the immune system is very immature. It's actually quite easy to trick. And the older we get, the cleverer our immune system gets as well. It stops at a certain age and we decline again. That's aging. So Duchenne boys have a slightly more mature immune system as well. And I also mentioned that these AAVs are affecting people even though they don't cause any symptoms. And the younger you are, the lower your risk that you had a viral infection. The older you become, the higher your risk that you already had been exposed and therefore have generated antibodies against the virus and can't be injected with gene therapy. It's also that we are now trying to actually just administer the gene therapy into the cerebrospinal fluid, into the central nervous system, which is limited in terms of an immune response. So it's more protected, whereas the systemic delivery makes the immune system more active. And this is important, and I'll come back to this later. Now it's also, if you think about your protein, the substance that the cell produces, for spinal muscular atrophy, it's called the survival motor neuron protein. And the way it is distributed in cells, it's in green here, is fairly diffuse, whereas dystrophin is a highly complex protein. It's like, I often compare it, it's often mentioned as a shock absorber, but that's the single protein. But as a network, dystrophin is like a stocking that you pull over a leg. It holds it together. It's also present at the so-called neuromuscular junction, where the nerve meets the fiber. It's also present at the so-called myotendinous junction, where the muscle fiber attaches to the bone. Now, at many conferences, you never hear about this. And companies also don't really specifically look at this. What does it do in gene therapy? There is the dystrophin at the myotendinous junction. How does it form this complex structure of what we call costamers, the stocking? So it's a lot more complex in Duchenne than it is in SMA. The SMN protein itself is very high around birth. So if you supplement it with gene therapy, perfect timing. Actually, later in life, we don't need much SMN. And it's also that every baby with SMA does actually produce some endogenous SMN as well. So the immune system is very familiar with SMN and the protein. There's no problem against the protein. Now in Duchenne, that's different. Dystrophin is expressed, as far as we know, throughout the life. Okay? You can't drop your dystrophin levels and say, oh, it doesn't matter. You always need a lot of dystrophin to protect fibers. And as I said in Duchenne, the immune system may not have never seen dystrophin, so it can attack a newly produced one. And then, of course, and we have heard a lot about this at the conference, Duchenne is a multi-system disorder. Whereas for spinal muscular atrophy, it is mainly the motor neuron that we need to target. In Duchenne, and I think we often underemphasize the importance of dystrophin expression in the heart. If we just express it in the skeletal muscle and not in the heart, we won't extend the life expectancy of patients with Duchenne muscular dystrophy. But we have heard a lot about the brain as well, and the gastrointestinal system. And so it's more complex in Duchenne again. So in summary, for spinal muscular atrophy, through AAV-mediated gene replacement therapy, babies with SMA type 1 can now be provided with a full-length SMN gene after birth, resulting in high levels of SMN protein in their cells. And therefore, the outcome of the gene therapy is actually amazing. No one has ever injected a patient with Duchenne muscular dystrophy with a full-length DMD gene at birth that resulted in high levels of dystrophin in skeletal and cardiac muscle cells. That experiment has never taken place. Then you can say, well, why not? Well, because there are additional challenges with dystrophin and the gene and its recipe, the RNA, because it's large. Dystrophin, the gene, is actually the largest gene that we know in nature, not just in humans, generally. There's no bigger gene that we know. And even its recipe, its RNA, is beyond the capacity of what you can put into an AAV virus. So therefore, if we compare this with a GPS system, you can only put something in that is called a mini or microdystrophin. Now, I also want to make this very clear that I personally think that this nomenclature is very confusing and bad, because it's neither mini nor micro what we put in there. It's about half the size, maybe a bit smaller. And so there's a nice table here that says a full-length dystrophin, of course, has perfect function. And then we have seen that there are patients with Duchenne muscular dystrophy, DMD, but also, and that is important, with Becker muscular dystrophy, a milder form of the same disease, where big chunks, and this is this delta, the deletion we say, are missing, where big chunks of the gene are missing. And the most famous one is probably this, what's called the deletion of exon 17 to 48. And so 46% are lost, but it means 54% are still there. It's not mini or micro, it's just a truncated version, okay? Now, because in Becker muscular dystrophy, in a milder form, we see that big chunks can be missing, we need to look a bit more into the dystrophin protein. So this is the gene. This is like the cookbook, as I said. And it's the information in those exons. This is the coding information, the information to make the protein goes from 1 to 79. And then the full-length recipe, the RNA, is 14 kilobases, so it's too big for the adeno-associated virus. And this is how the dystrophin protein looks. And everything that's blue here in the genetic information is basically represented in blue here in the protein. And in Becker patients, we know that it's normally these blue bits in the middle that can be missing, and you still have muscular dystrophy, but a milder form. And you can see this in families, and this was the famous family. It was actually a Newcastle family that Kay Davies reported first, where this deletion of exons 17 to 48. So if you go from here to here, this whole bit was missing. But the index case, which is this one, was still walking or lost ambulation at 61 years of age. And that's when scientists said, well, can we use this shorter form, the truncated form, and use this for gene therapy? And so this is the form of this patient. So it has dystrophin, but it's very short. And if you look at a muscle biopsy, these are antibodies in a normal, healthy muscle, and this is in a Becker muscular dystrophy. And this is against the middle part, and you can see it's missing. There's no dystrophin, so that middle part is missing. And then you can use from these Becker patients and can design genes, and this is what has first been done by Jeff Chamberlain's group, and then you can test it in mice and can see which one is actually the one that is working. And this delta 17 to 48 was one that was first trialed, and companies then said, okay, let's use these shorter dystrophins, put them in AAVs, and do clinical trials. And so these are some of the companies, and I'll talk more about the clinical programs, that have started to do this. And you can see that these are the dystrophins. They look fairly similar. There are subtle differences. And they have different promoters. So the ignition activate this inner tissue. And their vectors are not always the same. So there are differences, and that's good. It's good to have competitions to try and test. And we still don't know which of them is the best in terms of both safety and efficacy. So where are we now in the clinic? There is actually a lot more going on in terms of gene therapy. There are also some single-site studies in China and the US, sometimes also where patients with just a specific genetic change are treated. I'm not going to talk about these individual cases. But it's important to know that actually a lot of things are happening. But what's happening in the UK? And so Duchenne, the Duchenne Hub UK has a clinical trial finder. And so the clinical trial finder lists the trials that are happening in the UK. And this is what's currently in the clinical trial finder. And so one of the gene therapy trials, it's called EMBARK, and the sponsor is Sarepta Therapeutics, has already been completed. And I had talked about this, I think, last year and others as well. And you may remember that this was a trial that was supposed to show that after 1 year, the patients that were treated with the gene therapy compared to the ones that had a placebo, didn't get the gene, were supposed to show a statistically significant difference after 1 year in the North Star Ambulatory Assessment. This is the so-called outcome or primary endpoint. And what this bar shows you is that statistical significance wasn't met, and therefore formally the trial failed. What was encouraging is that the trend was towards the treated patients, and that there were other outcomes, assessment methods, that actually showed that the ones that were treated did show statistically significant improvement. So what was also encouraging is that there was dystrophin in the cells. The dystrophin, the percentage of dystrophin-positive fibers was about 30%, compared to almost nothing in the non-treated groups. And about 7 weeks ago, there was a paper published by Sarepta, supported by some clinical colleagues as well, like Francesco Montoni here in the audience. And they have now looked at, okay, how did the situation look 1 year after the injection? And 1 year after the injection, the patients that were treated with gene therapy compared to untreated patients showed all a benefit that was statistically significant. Now, the authors acknowledge that there is some limitation in this assessment because the patients that they used to control these results weren't patients of that trial. They were controls from other natural history studies. And as I said, the authors acknowledge that there are limitations for that method because you can have a selection bias. For example, the natural history study, some of them were years ago. So nevertheless, I think this is encouraging, and basically, if this is what the trajectory on average overall in Duchenne muscular dystrophy shows, this is age and this is your motor function based on the North Star assessment. What we calculate now is that there is a shift in the trajectory. Okay, that's what's currently, I think, encouraging. We need to now, I think, collect more real-world data, and the drug is approved in the US, not in the UK. Why not in the UK? Because it was initially approved based on dystrophin expression in the US. But as I said, the trial failed, and the European Medicines Agency said, well, the trial failed, therefore we are not going to provide marketing authorization. You will have also remembered or heard of the fact that some older Duchenne patients in another trial with the same gene therapy construct died. And because of this, the other Sarepta studies, the ENVISION study, is currently on hold to modify the protocol to just make sure that we have better safety monitoring. And there's a Roche study, and Roche has a license for that product in Europe, where London and Oxford are sites. This is currently on hold, probably also because of some safety issues, but probably also because they don't have marketing authorization in Europe, and there might be further delays. Okay, but there is a study that is recruiting in London. In Newcastle, we are still waiting to get the approval from Genethon, which is a company in France. And this is the full-length protein. They use, again, a truncated version. As I said, I don't like the term microdystrophin because it's quite big. They have a different promoter. It's the same as the Sarepta gene, but a different promoter, different ignition. They also have a different virus that they use, an AAV virus. So let's see, in this trial, patients started to be enrolled in a natural history study, and then there was a part 1 study where they tested 2 different doses of the gene therapy. The preferred dose has now been selected, and now currently the trial is recruiting patients actually in Spain, in the UK, in Belgium, and 64 patients are supposed to be enrolled. We need to wait. I can show you some preliminary results. Green is the patients that received gene therapy. Red is the control group. And so these are different outcomes. This is the rise from the floor. So it took the control group longer than the treatment group. The North Star was better. So everything looked as if there is efficacy, but these are preliminary results, so we have to now wait and see. But again, it's encouraging. This is just an acknowledgement to the team. Francesco presented these results also at the World Muscle Society. I haven't really mentioned the Pfizer trial because they stopped their program, and again, this was discussed here last year as well. Of course, the patients that were injected will be monitored for safety and continue to be monitored for this, but the trial has stopped. What I think is important to at least mention is that in terms of the amount of dystrophin in the muscle fibers, it was very similar to what we see in other trials. So we don't understand why that trial failed, even though dystrophin was quite abundant. So then there are trials from Solid. They are not yet recruiting, but they are planning to come to the UK. And so Solid uses, again, a construct that is slightly different, and their unique selling point is that they have some bits of dystrophin that's in the middle part that binds to another protein, which is called NOS, which might be relevant in the long term. They also have designed a virus that they hope will be taken up by the muscle fiber better. And they also have production improvements, making sure that the gene, the truncated dystrophin, is always in the virus, which isn't that simple, actually. Now, they have already recruited 33 participants that have been dosed between the ages of 1 and 10. And what was, again, very encouraging to see is that the safety profile was very good. And so, even that they can now administer this in an outpatient setting, and based on this, they're now going to do a phase 3 registration study to get marketing authorization. This is the overview of the study, and these are the eligibility criteria. This will be for boys between 7 and 12 years, and they need to be ambulant. They are planning to extend this to the UK. It's currently active in Australia and Canada. And so, again, great to see that this trial is coming to the UK. The pilot trials were very encouraging. This is the thank you slide from Solid Biosciences. And then there's another study that's currently ongoing in the States. Right now, I'm not aware that they want to come to the UK. If they get marketing authorization, and I mean, they will probably try to get this beyond the US, but the study is done in the US, and the company is called Regenex Bio, and they use an AAV8 vector, but it's also modified. And their unique selling point is that they still have part of one end of the gene, the C terminus, that none of the other companies have. That might be an advantage, we don't know. They also look at immunosuppression with new regimes, and also look at improving their manufacturing. And I think what is interesting here, and it's a bit of a learning from other trials as well, that in addition to steroids that everyone gets in gene therapy trials, they also have two drugs that are supposed to suppress this innate immune response, the immediate one, and the adaptive, the longer-term immune response. So they think through this, they give the virus and the proteins a better chance. And also avoid, of course, safety issues around depletion of platelets or liver toxicity or muscle and heart toxicity. Now, they have finished their pilot trial. The number of patients was very small. They saw more than 10% of dystrophin, and that's what they wanted to see. They had no serious safety events, and they're now, therefore, currently enrolling into their confirmatory study with 30 ambulant boys. And this is their acknowledgement slide, also thanking all the participants and families. Now, there's another trial going on in the States by a company called Insmed. And the study is called ASCEND. There's a difference here to all the other gene therapy trials in Duchenne muscular dystrophy. The age group is from 2 to 5, but they are injecting these boys into the cerebral spinal fluid, so by a lumbar puncture. Now, I mentioned before that in SMA, this is what we are doing now more and more, because the CNS is more protected from the immune system. The construct that they use is very similar to the one that Sarepta uses. They use a different AAV, but the same promoter as well, and it's currently in a phase 1, just looking at safety. And my understanding is that they have injected 6 patients so far. There's very little information about this so far. There's nothing published, but the 6 patients have been injected. And the big question, of course, for anyone probably is, but how does that help the muscle? How does a gene get into the muscle? I don't know. Again, they have shown that in mice this works also. I mean, you can go with your gene into the cerebrospinal fluid and dystrophin will pop up in the muscle. Now, again, I understand that there are also some scientific publications that are currently under review. So that was a summary about what's happening in gene therapy in Duchenne. And so what's coming next? Well, you have already seen from the trials that the trials are learning from each other. And that trials have specifically now focused on improving safety, so trying to trick the immune system. So there is more about immunosuppressive therapies, but it's also trying to modify the delivery system and even trying to replace a virus with a non-viral delivery method that might be less of a problem for the immune system. So attenuating the immune response is something that is happening. And I can see that ideally we want to achieve maybe redosing in the future. We're not there, but this is what's coming. Then the delivery, as I said, is the big challenge. And I think what we see with these better or new forms of either modified viruses or non-viral particles, that this might also affect capacity. And this is something actually, because you may all ask, where does AI come in? Because everyone is using AI. That's where AI is applied to. I mean, artificial intelligence can probably come up with better viruses or non-viral delivery forms. So many companies are starting to use this now as well. Just briefly, I mean, the perfect way would, of course, be to deliver a full-length dystrophin gene at birth, and if there's a dilution effect, to redose. That's what we want. I'm very confident that this will happen at some time, but it will take years. But there are now, and I said I'm not going to talk about mouse experiments, but there are now experiments where in mice we can see full-length delivery. And there are different strategies about this. Can you have smaller truncated forms of dystrophin that are then melted together or ligated, and then you have a full length? Or can you split up the recipe, the mRNA, and then put it together. So there are several concepts, and I think this is coming. So summarizing my talk, for those who didn't listen, it's the same slide as at the beginning. There's no approved gene therapy for DMD in the UK yet, but we have a good understanding of the concept, are becoming more familiar with the safety challenges and their management, and aim to further improve and confirm the efficacy through clinical trials and real-world data. I want to thank the amazing team at Newcastle for all the support. Want to thank you for your attention, and Emily and Alex again for the invitation. Thank you very much.